What is a common technique used for the amplification of DNA in biotechnology? A. PCR. B. ELISA. C. Gel electrophoresis. D. Western blot. The correct answer is A. PCR, polymerase chain reaction, is a common technique used for amplifying DNA in biotechnology. Which of the following is not a type of gene editing tool in biotechnology? A. CRISPR-Cas9. B. RNA interference, NA. C. ELISA. D. TALENS. The correct answer is C. ELISA is not a gene editing tool but rather an immunoassay technique used for detecting proteins. Which process involves the transfer of genes from one organism to another to confer specific traits or characteristics? A. Cloning. B. Mutation. C. Gene therapy. D. Genetic engineering. The correct answer is D. Genetic engineering involves the transfer of genes from one organism to another to confer specific traits or characteristics. What is the purpose of using recombinant DNA technology in biotechnology? A. To study ancient DNA. B. To create genetically modified organisms, GMOs. C. To synthesize artificial DNA. D. To analyze RNA sequences. The correct answer is B. Recombinant DNA technology is used to create genetically modified organisms, GMOs, with desired traits. Which technique is commonly used to determine the sequence of nucleotides in a DNA molecule? A. Microarray analysis. B. Southern blot. C. DNA sequencing. D. Northern blot. The correct answer is C. DNA sequencing is commonly used to determine the sequence of nucleotides in a DNA molecule. What is the function of restriction enzymes in biotechnology? A. Transcribe mRNA. B. Amplify DNA. C. Cut DNA at specific sequences. D. Generate cDNA. The correct answer is C. Restriction enzymes cut DNA at specific sequences, allowing for manipulation and analysis of DNA. Which technique is used to separate and analyze DNA fragments based on their size and charge? A. PCR. B. Western blot. C. Gel electrophoresis. D. Flow cytometry. The correct answer is C. Gel electrophoresis is used to separate and analyze DNA fragments based on their size and charge. What is the primary goal of gene therapy in biotechnology? A. To create genetically modified crops. B. To treat genetic disorders by replacing or repairing faulty genes. C. To clone animals. D. To study gene expression patterns. The correct answer is B. The primary goal of gene therapy is to treat genetic disorders by replacing or repairing faulty genes. Which technique allows for the rapid amplification of a specific DNA sequence in vitro? A. RT-PCR. B. Western blot. C. ELISA. D. PCR. The correct answer is D. PCR, polymerase chain reaction, allows for the rapid amplification of a specific DNA sequence in vitro. What is the role of vectors in gene cloning and genetic engineering? A. To transcribe genes into proteins. B. To amplify DNA. C. To deliver foreign DNA into host cells. D. To degrade RNA molecules. The correct answer is C. Vectors are used to deliver foreign DNA into host cells for gene cloning and genetic engineering purposes. What does PCR stand for in biotechnology? A. Polymerase chain reaction. B. Protein coding ribonucleotide. C. Plasmid cloning reagent. D. Polygenic cellular respiration. The correct answer is A. PCR stands for polymerase chain reaction, a technique used to amplify DNA in vitro. Which of the following is an example of a genetically modified organism, GMO? A. Banana. B. Tomato. C. Rice. D. Bacteria. The correct answer is D. Bacteria can be genetically modified to produce useful proteins or chemicals. What is the purpose of CRISPR-Cas9 technology in biotechnology? A. DNA sequencing. B. Gene editing. C. Protein purification. D. Cell culture. The correct answer is B. CRISPR-Cas9 is a gene editing tool widely used in biotechnology for precise modifications in DNA sequences. Which enzyme is commonly used to join DNA fragments in genetic engineering? A. DNA polymerase. B. Ligase. C. Helicase. D. RNA polymerase. The correct answer is B. Ligase is the enzyme used to join DNA fragments together in genetic engineering. What is the function of a plasmid in gene cloning? A. To transcribe mRNA. B. To translate proteins. C. To amplify DNA. D. To carry foreign DNA into a host cell. The correct answer is D. Plasmids are small circular DNA molecules used as vectors to carry foreign DNA into a host cell during gene cloning. What is the primary source of restriction enzymes used in genetic engineering processes? A. Bacteria. B. Fungi. C. Plants. D. Animals. The correct answer is A. Bacteria are the primary source of restriction enzymes used in genetic engineering processes. Which technique is used to create multiple copies of a specific DNA sequence in biotechnology? A. Gel electrophoresis. B. Southern blotting. C. Polymerase chain reaction. D. Northern blotting. The correct answer is C. Polymerase chain reaction, PCR, is used to amplify specific DNA sequences in biotechnology. What is the goal of gene editing technologies like CRISPR-Cas9 in biotechnology? A. To introduce mutations in DNA. B. To correct genetic disorders. C. To degrade DNA strands. D. To clone genes. The correct answer is B. Gene editing technologies like CRISPR-Cas9 aim to correct genetic disorders by modifying specific DNA sequences. Which technique is used to detect specific DNA sequences in a sample in biotechnology? A. Western blotting. B. Northern blotting. C. Southern blotting. D. DNA fingerprinting. The correct answer is C. Southern blotting is used to detect specific DNA sequences in a sample by hybridization. What is the significance of transgenic plants in biotechnology? A. They produce vaccines. B. They are resistant to pests. C. They secrete hormones. D. They generate electricity. The correct answer is B. Transgenic plants are engineered to be resistant to pests, reducing the need for chemical pesticide. What is the key function of antibodies in biotechnology assays and therapies? A. To indicate the presence of a specific antigen. B. To amplify DNA sequences. C. To induce gene editing processes. D. To initiate protein synthesis. The correct answer is A. Antibodies are essential in detecting and binding to specific antigens, indicating their presence in biotechnological applications. How are CRISPR-Cas systems commonly utilized in biotechnology? A. For targeted gene editing. B. For DNA amplification. C. For RNA sequencing. D. For protein purification. The correct answer is A. CRISPR-Cas systems are widely used for precise and targeted gene editing by modifying specific DNA sequences in various organisms. In biotechnology, what is the main purpose of RNA interference (RNA) technology? A. To silence specific gene expression. B. To induce DNA mutations. C. To stimulate protein production. D. To identify enzyme functions. The correct answer is A. RNA interference technology is utilized to silence or inhibit the expression of specific genes by targeting and degrading the corresponding mRNA molecules. Which method is commonly used for the separation and analysis of proteins based on their size and charge in biotechnology? A. Polymerase chain reaction, PCR. B. Western blotting. C. Gel electrophoresis. D. Restriction enzyme digestion. The correct answer is C. Gel electrophoresis is a technique used to separate and analyze proteins based on their size and charge by running them through a gel matrix under an electric field. What is the primary function of a gene gun in biotechnology applications? A. To deliver foreign DNA into target cells. B. To sequence DNA strands. C. To amplify DNA fragments. D. To analyze protein structures. The correct answer is A. A gene gun is a device used to deliver foreign DNA into target cells by propelling DNA-coated particles into the cells to introduce new genetic material. How do viral vectors contribute to gene therapy in biotechnology? A. By delivering therapeutic genes into target cells. B. By degrading excess DNA in cells. C. By detecting specific gene sequences. D. By replicating within host DNA. The correct answer is A. Viral vectors are commonly used in gene therapy to deliver therapeutic genes into target cells, allowing for the treatment or correction of genetic disorders. Which process plays a crucial role in the production of genetically modified organisms, GMOs, in agriculture and biotechnology? A. Gene silencing. B. Gene cloning. C. Gene transcription. D. Gene excision. The correct answer is B. Gene cloning is a fundamental process in the creation of genetically modified organisms, GMOs, by inserting foreign DNA sequences into the genomes of target organisms for specific traits or characteristics. What is the primary objective of bioprospecting in biotechnology research and development? A. To discover novel biological resources for commercial applications. B. To analyze genetic mutations in organisms. C. To synthesize artificial DNA structures. D. To quantify enzyme activity. The correct answer is A. Bioprospecting aims to discover and evaluate new biological resources from various natural sources for potential commercial applications in biotechnology and pharmaceutical industries. How are stem cells utilized in regenerative medicine applications within biotechnology? A. To differentiate into various cell types for tissue repair. B. To increase DNA replication rates. C. To induce gene mutations. D. To regulate protein synthesis. The correct answer is A. Stem cells are used in regenerative medicine to differentiate into specialized cell types and repair damaged tissues, offering potential therapeutic solutions for various medical conditions and injuries. Which technique is commonly employed for the detection and quantification of gene expression levels in biotechnology research? A. Real-time PCR, polymerase chain reaction. B. Gene cloning. C. Western blotting. D. Plasmid transformation. The correct answer is A. Real-time PCR is a widely used method to detect and quantify gene expression levels by monitoring the amplification of DNA samples in real time, allowing for precise measurement of gene activity. What is the principle behind DNA fingerprinting in biotechnology? A. Unique sequence of DNA in each individual. B. Identification of specific genes in a DNA sample. C. Amplification of DNA in vitro. D. Sequencing entire genomes of organisms. The correct answer is A. DNA fingerprinting relies on the fact that each individual has a unique sequence of DNA, which can be used for identification purposes. How do plasmids contribute to gene cloning in biotechnology? A. Carry foreign genes into host cells. B. Amplify DNA segments using PCR. C. Restrict the expression of inserted genes. D. Isolate specific DNA sequences from a genome. The correct answer is A. Plasmids are small, circular DNA molecules that can carry foreign genes into host cells, aiding in gene cloning and recombinant DNA technology. What is the role of DNA ligase in genetic engineering processes? A. Cuts DNA at specific recognition sites. B. Adds new nucleotides to a DNA strand. C. Joins DNA fragments together. D. Ensures accurate replication of DNA. The correct answer is C. DNA ligase is an enzyme that seals nicks in the sugar phosphate backbone of DNA strands, joining DNA fragments together during genetic engineering processes. In biotechnology, what is the function of RNA interference, NA, technology? A. Detecting specific DNA sequences. B. Sequencing entire genomes. C. Regulating gene expression. D. Studying protein-protein interactions. The correct answer is C. RNA interference, NA, technology is used to regulate gene expression by targeting specific mRNA molecules for degradation, affecting gene silencing and protein production. Why are transgenic animals used in biotechnology research? A. To generate genetically modified crops. B. To study gene function and disease models. C. To analyze protein-protein interactions. D. To determine DNA fingerprint profiles. The correct answer is B. Transgenic animals are used in biotechnology research to study gene function, develop disease models, and investigate various biological processes through genetic manipulation. What is the significance of CRISPR technology in gene editing and biotechnology? A. Detecting gene expression levels. B. Editing specific DNA sequences. C. Isolating genes from a genome. D. Producing genetically modified organisms. The correct answer is B. CRISPR technology allows for precise editing of specific DNA sequences, enabling targeted modifications in genomes for various biotechnological applications. How do Southern blotting techniques contribute to DNA analysis in biotechnology? A. Detecting protein interactions. B. Sequencing RNA molecules. C. Identifying DNA fragments. D. Amplifying specific gene sequences. The correct answer is C. Southern blotting is a technique used to identify specific DNA fragments in a mixture, aiding in DNA analysis, gene mapping, and understanding of genetic variation in biotechnology. Which technique is commonly used for the separation and analysis of proteins based on their size and charge in biotechnology? A. Polymerase chain reaction, PCR. B. Western blotting. C. Gel electrophoresis. D. Next generation sequencing. The correct answer is C. Gel electrophoresis is a commonly used technique for separating and analyzing proteins based on their size and charge in biotechnology research and diagnostics. What is the primary purpose of using gene editing technologies like CRISPR-Cas9 in biotechnology? A. Detecting protein-protein interactions. B. Producing genetically modified organisms. C. Editing specific DNA sequences. D. Studying gene expression levels. The correct answer is C. Gene editing technologies like CRISPR-Cas9 are utilized in biotechnology to selectively edit specific DNA sequences, allowing precise modifications for various applications such as gene therapy and genetic engineering. How do viral vectors contribute to gene therapy in biotechnology? A. Enhance protein synthesis in host cells. B. Deliver therapeutic genes into target cells. C. Detect specific DNA sequences. D. Amplify gene expression levels. The correct answer is B. Viral vectors are used in gene therapy to deliver therapeutic genes into target cells, enabling the correction of genetic defects and treatment of various genetic disorders in biotechnology applications.